Welcome back, mitochondriacs. It's time to talk about some red light therapy. What do you say? This happens to be probably one of my favorite topics. Now, you weren't early, I'm late. All right, let's see if I can get this pulled up here. Give me 1 second here. Goodland, Kansas. That's a long ways away. I'm from Topeka originally. Even that's a long ways from me, and I used to live up there. I was doing some research on exercise yesterday before I wanted to present this, and I had to go and find the right slides. So give me 1 second here. Handheld red light. All right. So I think that the best way to discuss photobiomodulation or the use of light to affect our biology is by starting with the electromagnetic spectrum. And I'm not going to sit here and pretend that I know everything about this science, but what I will say is that, the sun puts off all kinds of different waves, but some of those can be taken up from the ionosphere and the atmosphere and so on and so forth. And what we get left with is kind of like ultraviolet, visible, and infrared. And you're going to see nomenclature when we start talking about specific parts of the spectrums, and it's going to be listed as nanometers, okay, nm, and that's the actual length of the light photon. And so if it doesn't show it here in this graph really that well, but the ultraviolet would be like 250 nanometers to about 400 nanometers, and then that's broken into UV A, B, and C. And UVA is what tans us and activates POMC and different things. UVB is how we make vitamin D, and UVC. I don't know if we know a biologic use for it yet. I think that if anybody would know, Dr. Cruz would know. And then we have the visible, of course, violet, blue, green, yellow, and red. And then we have infrared. And infrared is also broken down into 3 different types of wavelengths. We have near infrared or infrared A, mid infrared or infrared B, and then far infrared or infrared C. And each kind of like little grouping of infrared A, B, and C will have a very specific wavelength and where it starts and where it ends. And so when people talk about like near infrared light therapy or far infrared saunas, they're both invisible, light, but to the naked eye, but they mean completely different things. And those different things can have vastly different biologic activities. So just to give a different way of looking at it, we have, and just to be very clear, this is like flipped on its side. So you can see that gamma is on the left here, and on this side, gamma is on the right. But I'll digress here. So we have gamma rays, we have X-rays, we have ultraviolet, we have the visible, we have infrared, and then we have other waves, right? Like microwaves, and microwaves are its own thing where there's like ultra low frequency, high frequency. And, if we ever get into a chance to talk about EMF, we can go into those more specifically. But going back to the sun spectrum, the sun puts out somewhere like 260, 270, all the way to about 3,000 nanometers. And the vast majority of the solar spectrum, in fact, 52% is that near infrared. And that is what's available, most of the day. So to be fair, this is looking at the tropics during summer solstice. But that being said, you'll see that infrared A, okay, is available all the way from sun up to sundown, basically, okay? And it's obviously going to be more intense throughout the day as the angle of inclination is different and you have a stronger kind of like beam towards you. But, that's what you'll see. It's 30-fold higher at noon than it is at 6 a.m. But the bottom line is it's present. So, some people will say, oh, you got to get your infrared in the morning. Well, you can get it in the morning, depending on your location and the time of year, but you can also get it at noon, and you're going to get it just like vitamin D. You're going to get the biggest bang for the buck at solar noon when the sun is the strongest. Yeah, so Axis 2 is asking some questions about, or not really asking questions, she's kind of giving me some suggestion. And I'm going to talk a little bit about easy water and stuff like that. I don't pretend to be an expert, but I do want to talk about the major biologic mechanisms that have been proposed for red light therapy and its actions on cytochrome C oxidase. And what it says here is the primary site of light absorption in mammalian cells has been identified as the mitochondria, and more specifically cytochrome C oxidase. It is hypothesized that the inhibitory nitric oxide can be dissociated from CCO, thus restoring electron transport and increasing mitochondrial membrane potential. And there are probably thousands of papers talking about red light therapy and all the different things that it can do. But if we go back to the basics of mitochondrial electron transport, we can see that ultimately foodstuffs in the form of proteins, carbohydrates, and fats are donating protons and electrons through NADH and FADH2, and the protons get into the inner mitochondrial matrix and build a gradient, and the electrons are transported through the electron transport chain so that ultimately we can actually make ATP, right? That's the classic way of looking at energy production in the body. Now, there are potential problems with that as being the only way to get energy. I agree fully with what Gilbert Ling and Dr. Jack Cruz, who's been a champion for this, that there's probably other ways in the body where energy is made, but there's no question that we rely, heavily on ATP and the electron transport chain. So what red light does is they hypothesize is that it will liberate one of the blockers of cytochrome C oxidase, nitric oxide, and it will allow electrons to transport through cytochrome C oxidase more effectively and more efficiently. Now, there are many other modes of energy production that are proposed, or I shouldn't say energy production, but there are other models of how red light works. And I'm going to touch on those. But ultimately what we have is that we have infrared light or visible red light. When we talk about photobiomodulation, it technically does mean any wavelength of light having a biologic effect. But to be more fair, it's generally talking about red light and infrared light, and specifically near infrared red light. And you see here that this irradiation is going through the eye, in this case, is affecting the mitochondria. It's affecting the electron transport chain. That's going to have what they call retrograde signaling, where you have basically signals from the mitochondrial electron transport chain, likely ROS, RNS, that go and talk to the nucleus, and the nucleus will then alter its gene expression and ultimately protein synthesis, which will then create a more suitable environment for the cell itself. And this is just kind of like another way of looking at it. And you'll see here that sometimes it's labeled. Yeah, these just say light beams, but in this case, it actually says 830, which is in the near infrared range. and it's having that effect, like we mentioned, on cytochrome C oxidase, which then changes all kinds of different things within the cell, basically. And we believe that those things are favorable, as long as it's within reason. And that within reason is still being determined. There are biphasic responses and so on and so forth. I think it's probably more likely to be a thing within red lights from an LED or a laser than it is the sun, because I think that we were designed to be out in the sun all day long. So if we had biphasic responses, we would have negative effects of being in the sun. And I don't think that's going to be the case. But when you're shooting a light or a laser, then I think it makes more sense. So this is where I want to talk about the other things that light is able to do. And it takes us back a little bit towards water, okay? So this is a paper that was written by Mei-Wan Ho in 2015, who has passed away, as well as Emilio Del Giusse. He's also passed away. But what they have, and when I mean they, I mean physicists who are really good with, numbers and calculations and stuff like that. And they've figured out that there's a discrepancy of some degree of magnitude where ATP does not make up for that discrepancy. And so they hypothesize that there are other ways that energy is made. And what Del Giusse and his colleagues is it said the interaction with light and liquid water generates quantum coherent domains in which the water molecules oscillate within a ground state and an excited state close to the ionizing potential of water. This produces a plasma of almost free electrons favoring redox reactions, the basis of energy metabolism in living organisms. So this is a much different way of looking at how energy is produced within a cell. And it says here that the coherent domains is thus a redox pile where almost free electrons can be readily donated to electron acceptors. Excited coherent domains is the basis of oxidation reduction energy metabolism that powers all living processes. It is both the chemistry and electricity of life, and therefore it orchestrates all necessary chemical reactions. And I don't think I need to potentially go over every single one of these slides that I produced, but what I do want to show is how it ties into research that was done later by a researcher named Gerald Pollack at the University of Washington. And what they were able to find was essentially the same, how do I want to say it? They found the same information, but from a different way of looking at it. And so what Del Giusse and colleagues and Mei-Wan Ho referred to this as coherent domains, Dr. Pollack refers to it as exclusion zone or EZ water. And basically what they were able to do is charge separate water. Water has O and H, right? H2O. And so the H's are positively charged and then the water and the oxygen is negatively charged. So when under certain circumstances, the water will line up in a very orderly fashion, not random at all, very orderly. And it has to be next to what they call a hydrophilic surface. And then when they are able to take electrodes, they are able to actually light up a battery or light up a light bulb because of the electrochemical gradient that's formed. And what they were able to find was that water is able to absorb light all the way from 250 nanometers all the way to more than 3,000 nanometers. Okay, so the entire spectrum of the sun is absorbed in water. And it's a very special property that water has. And what they were able to find is that if you shined ultraviolet light at 270 nanometers, which is right on the cusp of UVA, well, no, that would be UVB, excuse me. And when you're shining UVB onto water, the coherent domains, or the EZ, as Dr. Pollack calls it, grew by 40%, essentially 1.4 times. And then when they shined visible red light, it went 2.2 times. And then they went to far, middle infrared and far infrared, and it grew as high as 3 times. So what they're able to find is that when you shine light on the water, that's all, we always talk about, the human body is 60% water, 50% water, 70% water, and it depends on your age. But my point is that water is not inert. That water has biologic function, and that, mainstream science has not caught on to that, or don't necessarily believe that. They believe it's just an inert, kind of aqueous substance where chemical reactions are happening. But if you take into the account this kind of science, you see that is not the truth. And you also see that life is taking advantage of this positive and negative charge that is built up when you're exposed to light. So all that to say that there are a lot of researchers that are looking at water and photobiomodulation to see what also could be happening. And I think that there's some pretty cool graphics, like this one potentially looking at, UV, visible, IR having an effect on water. And then, that's how charge separation happens, and energy storage at some level, changes in water density, and how that affects gene expression and affects diseases, right? And so it's pretty cool. And I think I'm going to probably conclude this portion of the, interesting talk that I don't have really business being very frank with you, like teaching from a high level, just because this is above and beyond my understanding. But that being said, is that they do believe that red light will lower the viscosity of water, and that will allow not only ATP through the cytochrome C oxidase mechanism that we talked about in the first couple of slides, but it will also allow the ATPase to spin much easier and more freely, and able to produce more energy via that way. And then when you're exposed to, and this is an illustration of being outside in nature in the sun, being exposed to red light, having a lot more ATP, and then you see that high viscosity when you're in the city, when you're exposed to blue light mostly, then that damages the ability for the ATPase, or the nanomotor that makes ATP, to be as efficient. And I just think it's very interesting. I just want to touch on it at least, because I think that I know I'm going to get comments on it later. Oh, what about Dr. Pollock's work? Or, oh, what about this? And I wanted to make sure that I at least touch on it, not necessarily from an area of expertise, but at least that I'm aware of it, and that I know that there's more than just what I'm presenting as benefits. So with that, I'll digress. I'm just going to read one question. I'm attempting to restore OXPHOS, reverse Warburg effect, in cancer cells by consuming methylene blue as opposed to, yes. So we're going to talk about photodynamic therapy, probably not during this live stream, just because we have a lot to cover, but I will talk about photodynamic therapy as well as methylene blue. I'm not going to leave anybody hanging with that. Okay, so I've had many other comments from people that have taken my course, that I've had in consultation, and via the comments. Is photobiomodulation, or the use of red and infrared light therapeutically, is it bad if I have cancer? Because if you Google search it, one of the first things that comes up is that, I think it was an oral squamous cell carcinoma that was potentially made worse by red light. And I want to kind of take an axe to that, and I want to break down the one paper, that explains that, and hopefully give us some good news here. So this one's called Photobiomodulation in Cancer. What is the truth? And it says that although there are a few articles suggesting that PBM, or photobiomodulation, or red light therapy, can be detrimental in animal models of tumors, there are also many articles that suggest the opposite, and that light can directly damage the tumor, can potentiate other cancer therapies, and can stimulate the host immune system. Moreover, there are two clinical trials showing increased survival in cancer patients who received photobiomodulation therapy. So I'm going to start out with probably where, I would say that probably where the majority of medicine would like to keep PBM is just as a way. It's kind of like acupuncture or yoga or meditation. They want to they don't want to say or look at whether or not it helps cancer, like the tumor gets smaller or people live longer. What they have, at least what I've seen in the literature with these like alternative therapies or with these, integrative therapies, is they want to look at how I can help the cancer patient like have a better quality of life, which I'm not saying that's bad. Not at all. As a matter of fact, if I can improve your quality of life, that's amazing. But I want to look at it to see whether or not this has an effect on cancer itself, right? So I just want to show here that, they've looked at it from, reversing potential brain damage after glioblastoma, probably surgery or radiation therapies, improvements in quote unquote chemo brain, oral mucositis, as there's a lot of literature for oral mucositis, potential cardiac toxicity. Some chemotherapeutics like I want to say doxorubicin, dianorubicin have cardiac toxicity. So therefore, maybe there's a need for photobiomodulation and kind of regeneration after using that. Peripheral neuropathy is big. Wound healing, any kind of like radiation fibrosis, and any other toxicities that are associated with chemotherapy or treatment-related, side effects of radiation as well. Photobiomodulation has been shown and studied in many different models to show benefit. But I want to cut past that and look at like what about photobiomodulation's effect on cancer and where do we stand, right? So despite the existence of numerous studies that have shown PBM can increase the growth of cancer cells and cell culture, the number of studies that suggest that PBM can actually exacerbate or stimulate cancer growth in animal models in vivo are relatively few. One study compared the effect of PBM, this is a red light study, 660 nanometers, which is right in the middle of the red light range, visible red light, delivered once a day for 3 days, either at a low dose or at a high dose in subcutaneous melanoma in mice. The low dose, 150 joules per centimeter squared, reduced the tumor size, not statistically significant, while the high dose, 1,000 joules, significantly increased the tumor size. However, this study suffered from some problems, such as the claim that this type of tumor was grown in a non-synergistic mouse strain. So I think this is kind of interesting because the low dose, they're saying, reduce the tumor size, not to a statistically significant amount, but the higher dose increased the tumor size. And they were saying that, well, the reason was because this tumor, this is like a created tumor that was in a non-synergistic mouse strain. That, I'm guessing, could be a fatal flaw to that potential study. Another study looked at 650 nanometers, again, right in the middle of the red range, as a single dose on this mouse model of this anaplastic or very like highly disorganized thyroid cancer. And these investigators used an immunodeficient nude model, which does not accurately reflect human patients. So what the author of this particular paper, this paper here, is saying that they're kind of like picking apart these negative studies and kind of explaining how that really wouldn't be the case for a good cancer model, nor for a cancer model that really represents humans. So I think that's kind of interesting. Then on the complete flip side, they say, well, can PBM or photobiomodulation directly or indirectly attack cancer? And it gives us several mechanisms, proposed mechanisms. Okay, it says the first involves a direct effect of the light on the tumor cells themselves, and may be thought of as a deliberate use of the biphasic response curve to overdose the cancer cells. So within photobiomodulation, there are response curves, and there is the thought that you can actually overdo red light therapy. And basically what they're saying is they're using this response curve as a way to overdo the red light therapy for cancer cells in particular. And it was saying that a single dose, 1,200 joules centimeter squared, caused complete regression of tumors, which did not occur in these tumors that are lacking functional mitochondria. So what does that essentially mean? Essentially, one of the mechanisms that we kind of mentioned here was that when you irradiate something with whatever light we're using, so we're using, let's say 650 nanometer visible red light, you're going to increase ATP production within functional mitochondria, and you're going to actually increase reactive oxygen species to a small degree, or the right degree, that alters gene expression and then makes the cell stronger. Okay. However, when you have cancer cells that have either absent or non-functioning mitochondria, then what happens is that you actually create reactive oxygen species bursts, which terminally damages the mitochondria and creates an apoptosis response, or the reactive oxygen species are so overwhelming that it will cause DNA damage and cause apoptosis that way. So again, they gave a fairly large dose, 1200 joules per centimeter squared, but one time, and it caused a complete regression of the tumor. But it didn't happen within tumors that were lacking functional mitochondria. So it was likely the cytochrome C caspase apoptosis response from these lacking functional mitochondria. Are we keeping up here? So we got a couple comments here. So nuclear spin is affected by light. Yes, I'm sure that it is affected by light. I have to be honest with you, I have no way of really talking much on a high level with that. I've listened to Jack Kruse for years and years, and I just don't have the physics knowledge, to be very frank with you, to talk about that kind of stuff. Yes, water is amazing. There are over 30 types of ice. Yes. Yep. Yeah. So this is this access to, you're right, you're spot on. And as a matter of fact, this is a deeper question, right? So I've had this question several times. If I give melatonin and restore energy flux through mitochondria, am I helping the cancer? If I give DCA, if I give, thiamine, if I give whatever it is, and I restore energy flux through mitochondria, will I not be helping the cancer? And the answer is no, likely not. They believe that when you restore electron flow through a damaged mitochondria, you then restore apoptosis, which has been basically inactivated by cancer cells. So that's why I am more bullish on photobiomodulation and red light therapy for cancer. And I don't want to say I disregard the negative studies. I look at them, but mechanistically, it would make more sense to have an inhibitory or a helping effect by red light therapy than it would have a harming effect. So the second method that they hypothesize is that it's relying on taking advantage of differential effects of PBM between malignant cells compared to those seen in normal healthy cells. This involves combining photobiomodulation with additional cytotoxic anti-cancer therapy so that it increases the killing of cancer cells while at the same time protecting normal healthy cells. These considerations are related to the Warburg effect, by which the mitochondria of cancer cells change their metabolism to carry out aerobic glycolysis instead of oxidative phosphorylation. This phenomenon occurs due to the rapid growth of tumor cells outpacing the development of sufficient blood supply, forcing the cancer cells to become tolerant to chronic hypoxia or low oxygen levels. Glycolysis consumes much less oxygen than oxidative phosphorylation. The consequences of the Warburg effect are that malignant cells and normal cells may behave very differently in response to PBM. This is a key point, okay? So just like a lot of other things we've seen, whether it be melatonin or vitamin D or EGCG or quercetin, we see that it has one effect within cancer cells and it has almost an entirely different effect within healthy cells. And they're saying that this is likely the case with photobiomodulation as well. In cancer cells, where ATP supply is quite limited, the ATP boost given by PBM may allow the cancer cells to respond to pro-apoptotic cytotoxic stimuli with more efficiently executed cell death apoptosis programs, which are heavily energy dependent, require a lot of ATP. In contrast, normal cells have an adequate supply of ATP. The effect of PBM produces a burst of ROS that could induce protective mechanisms and reduce the damaging effect of cancer therapy on healthy tissues. So not only is it not damaging to the normal cells, it actually improves their ability to tolerate potentially like radiation therapy or chemo or whatnot. So it's kind of a double whammy. And my thought is that it's probably having an effect in a variety of ways. I don't like relegating it to this mechanism or this mechanism or this mechanism. I think that restoring ATP is part of it. I also think that creating overwhelming amounts of ROS is also part of it. And that's kind of what it's showing here. It's just showing that in a normal cell, protective mechanisms are induced, ATP is increased, ROS is increased, normal mitochondria. This is what you got. Within a cancer cell, ATP is actually fairly low because it's using aerobic glycolysis, which is much less inefficient or much less efficient. And when you give this ATP boost, quote unquote, I think it's going to be more like an oxidative stress boost. It doesn't show it in here, but you're going to see more of an ROS boost. That's what's going to restore the apoptosis, because if you know anything about mitochondrial biology, it's this cytochrome C. When it's ejected from the mitochondria, that's when apoptosis is allowed to be restored. So what we're doing is we're creating a bunch of extra reactive oxygen species by stimulating cytochrome C oxidase within a very broken mitochondria. And just to like be even more clear, if you look at Dr. Seyfried's work, when they study mitochondria and they look at the structure of mitochondria under the electron micrograph, you'll see that healthy mitochondria will have beautiful cristae, and those cristae are critically important for mitochondrial function and to reduce reactive oxygen species production. And so when you have damaged mitochondria, yes, you may be putting some more electrons through the electron transport chain, but you're going to be so inefficient at doing that, and you're going to produce a bunch of reactive oxygen species, which is going to ultimately cause like a ferroptosis basically within, or a lipid, kind of more likely a lipid peroxidation and ferroptosis within the mitochondrial membranes, and allow cytochrome C to be exited and then create the apoptosis stimulus we're looking for. It's very similar to, I'm going to try to scroll down here and find it because I have, if you can imagine, a lot of slides here, but I've already located it. So this slide is synonymous, and it's the same with many other things, okay? When we take the brake off of PDK, okay, and we allow flux of energy to go through the mitochondria, we see that it creates reactive oxygen species, DNA damage, and cell death. Why is that? Because these mitochondria are damaged, okay? These mitochondria are not able to handle that kind of basically energy going through them. They are not capable. So I believe that PDK, even though it's pathologic, it's likely a protective mechanism against cancer because it helps protect themselves from reactive oxygen species, and it helps shut off apoptosis. And DCA, dichloroacetate, it unlocks apoptosis via that mechanism. Same substance, different graph. Unlocks apoptosis through ROS, and by putting electrons through the electron transport chain that is damaged. So my belief is that probably the strongest mechanism for PBM is going to be its oxidative stress that it produces on these cancer cells. I'll digress. There's a third mechanism that is proposed by these researchers, and it's saying that in which PBM could be beneficial to cancer patients is possible role in stimulation of the immune system to fight the cancer. They showed in this particular model of melanoma, this mouse model of melanoma, where they were giving different wavelengths of light and different amounts. So they used 660 nanometers, which is red light at 50. This is an irradiance amount, 50 milliwatts per centimeter squared, 3 joules. 800 to 970 nanometers, which is in the infrared A range, at a much higher irradiance for double the amount of joules, once a day for 4 days, could all reduce tumor growth and increase the recruitment of immune cells, in particular T lymphocytes and dendritic cells secreting type 1 interferons. PBM also reduced the number of highly angiogenic macrophages within the tumor mass and promoted blood vessel normalization, which is another strategy in control of tumor progression. So this is pretty astounding, right? We have one light that is able to kill cancer cells directly through excess reactive oxygen species, is going to help restore apoptosis through improvements in energy flux through mitochondria, and potentially handling the energy deficit within cancer cells that they can't activate apoptosis. I have a question about that one. And then the 3, improving our our immune system against cancer cells. And one thing I'll say is that if you notice these studies, okay, how many of you who have red light panels out there I'm gonna buy a $1,000 red light panel, and I'm gonna use it once, and I'm gonna call it good. I'm good now. So even the studies that what they're using is like kind of unrealistic, you know, what we could have available, right? Like they're using this once a day for 4 days. Okay, they're seeing like this amazing responses. Why not use it more in line with how people would use it in real life, right? Like maybe they would use it 20 minutes a day or 40 minutes a day, you know, or 10 minutes a day, but daily for a longer period of time. This is what really frustrates me about conventional literature and research, is that they'll use something that like likely works or likely has an effect, but then they'll like underdose it, or they'll like do like a one-time dose. And it's just not realistic that you're going to have the outcome you're looking for. Like we're having amazing outcomes just with like very minimal inputs. So like, I'm not trying to like negate that we're already seeing benefit with like 1 or 4 days. But the bottom line is this could be used for the rest of your life. It could be used daily. It could be used several times per week for the rest of your life. You know, it's unrealistic, you know, and it's not like you give let's say we're using, I don't know, a chemotherapeutic, we're using doxorubicin. It's not like you do a study like, okay, we gave one dose of doxorubicin and the cancer's not gone. Like, yeah, it's going to take like multiple cycles of this chemotherapy for it to be effective. Like, and you study it with that, like, why don't you study it with these kind of things? And I think it's just probably the industry protecting itself, to be honest with you. I don't think that they really want to show efficacy, or at least like magical efficacy, like with these kind of therapies, because this would take over as the main therapeutic strategy going forward, right? And it would collapse the other industries. But I'll digress. So this is another paper that's called Reviewing the Efficacy of Photobiomodulation Therapy in Oncological Treatment, and it's published very recently, October 8th of 2024. And what they did is they essentially broke down a lot of the studies that are on photobiomodulation. And in this case, they're using lasers. And in this next one, they're using LEDs at varying wavelengths. You can see that some of them are even in the blue range. It's all over the place, to say the least. But what I'm going to double down on is that, look, the protocol is a single application. Okay, this one had daily, you know, but a single application. They all say single application. And you go to the LED studies and you see the same thing, single application. Okay, this one got 3 applications, you know, and then it showed the study conclusions. They're able to make a conclusion completely about red light therapy when you're studying it with one single application, or maybe 3 or 4 applications. And if you look at the ones, even the single application inhibited growth of melanoma cancer cells. I'm just going to go down here. Decreased proliferation, decreased melanoma cell viability, direct anti-tumor effects on these colon cancer cells or melanoma or whatever, right? Single application. If you look at the one study they did here where they had 3 applications, PBMT or photobiomodulation therapy at high doses inhibited the progression and number of oral squamous cell colonies. So I think that although like there's been a lot of research on cancer with red light therapy or photobiomodulation therapy, it needs to be like completely like done in different ways, in my humble opinion. They need to be doing it over like weeks and months and seeing what kind of like effects you're going to have in these models. But I do think it's interesting, though, that being said, that they have multiple kind of like potential mechanisms of how photobiomodulation can actually work. And I think that this can potentially show us, what is possible within the red light therapy realm. And I just want to make sure that I'm not getting down into some other things here. So, okay. So I'm going to end on this because this is starting to get into some combination therapies, and I want to really cover today only on red light therapy by itself. But that being said, as a teaser, I'm going to give some other things. So this is in the same paper here, reviewing the efficacy of photobiomodulation therapy in oncologic treatment. And it says here that there are combination therapies, and the combination therapies, they showed like they used certain sensitizing molecules with red light therapy. And one of the interesting molecules that they used in this particular in this breakdown of this paper was that they used in a melanoma model, a breast cancer model, and normal fibroblasts and breast epithelial cells. They shot 660 nanometer light via a laser, which is right in the red range, and they used a compound called gallic acid. And they wanted to see, what was the effect of gallic acid by itself, and then what would be the effect of gallic acid when followed by this red light therapy. And so the pretreated cancer cells with laser first, then gallic acid reduced viability more than gallic acid followed by a laser. The laser plus gallic acid combination increased reactive oxygen species production, apoptosis, and ferroptosis in cancer cells compared to gallic acid alone, while sparing normal cells. This suggests that low-dose laser can sensitize breast and melanoma cancers to the cytotoxic effects of gallic acid via ROS-mediated cell death pathways like apoptosis and ferroptosis, representing a potential therapeutic approach. And I'm going to have to move this out of the way because I can't see. And then it says later on, it says that the combination of PBMT with other treatments appeared to offer synergistic effects that could be leveraged for more effective cancer therapies. And I'm gonna end on what it says here. It says that the studies suggest that PBMT, photobiomodulation therapy, does not interfere with the effectiveness of traditional cancer treatments like radiation therapy, and may even inhibit cancer progression by affecting cellular pathways involved in migration and invasion. These findings indicate that photobiomodulation or red light therapy could be incorporated into existing treatment regimens without diminishing their efficacy. So I want to really hammer that point home, because one of the I don't think it's done out of malice in most cases, but one of the things that's very irritating that I hear on a regular basis is that oncologists will tell patients that if you do IV vitamin C, or if you do HBOT, or if you do this ketogenic diet, or if you do this, let's just say red light therapy, it's going to negate our chemo or radiation strategies, or they'll say it'll do nothing at all. And I don't know where they're getting that information from, the majority of the time. So my guess is that they know what they know, and they know they unfortunately they don't know what they don't know, and they will tell patients that out of caution that it's going to harm the treatment as a scare tactic. But then the patients who are willing and able to get onto research, publications and to the National Library of Medicine and published literature, they can see that things like this, right? And it creates a discrepancy. So first recommendation is oncologists and anybody shouldn't talk about things that they don't know about as a matter of fact, okay? If you don't know, just say you don't know. But in this case, it looks like for at least red light therapy, it likely enhances and does not interfere with the standard of care. Okay. Now, the same is true for HBOT and IV vitamin C, as long as it's the right dose of vitamin C and many other things. They tell people that. But my point is that if you've caught up with our work at all, okay, in terms of like how we're laying out metabolic therapy and how it functionally works, we're restricting glucose. That's going to restrict ATP. That's going to restrict lactic acid. That's going to restrict pentose phosphate pathway metabolites, which is going to restrict nucleic acid and NADPH synthesis, which is going to hurt the cancer cell's ability to recycle glutathione. Okay, it's going to make them more sensitive to oxidative stress. By restricting glutamine, we're doing the same thing. We're actually blocking the cancer cell's ability to actually make glutathione, because glutathione requires glutamate, requires cysteine, and requires I'm blanking on the name. But my point is it requires glycine. It requires 3 amino acids in order to make glutathione. So by blocking glucose and glutamine, you are basically shutting down energy production, yes, but you're also shutting down these cancer cells' ability to protect themselves from oxidative stress, okay? Which is why we spent so much time talking about ferroptosis, SLC7A11, which is that glutamine cysteine antiporter, and how it has been shown to be like the Achilles heel of cancer, right? Because it is so heavily reliant on that cysteine coming in and that glutamine being there so that it can make glutathione and protect itself against its own redox reactions and redox chemistry, but also other things that we try to do to it, whether it be radiation, chemo, photobiomodulation, whatever it is, right? So it essentially has the shields up with all the extra glutathione around. When we shut down glucose and glutamine and we lower the shields, then cancer cells in general are much more susceptible to oxidative stress. And we know from this particular, live stream and this lecture today that one of the main proposed mechanisms of how PBM likely is very effective at killing cancer cells and helping our normal cells, is that it creates massive amounts of reactive oxygen species within the damaged mitochondria of cancer cells, not within healthy mitochondria. So I think that you can see that this could easily dovetail into our purposes here with metabolic therapy as a whole, okay? And it's going to get better, because when we start talking about photodynamic therapies, by using photosensitive molecules, in this particular paper, they talked about gallic acid being synergistic with red light therapy. There are other molecules that can be synergistic with red light therapy, like methylene blue, okay, which is what someone else had brought up. So I want to talk about photodynamic therapy and its own thing, all right? However, you can start to see how red light therapy and photobiomodulation can dovetail into metabolic therapy. Now, if you're still nervous about using red light therapy because there are a couple like very small papers showing that it can make things worse, okay, what I tell people who have that nervous tendency, which I totally understand, that I would use photobiomodulation in a different way, okay? And that different way is how I want to talk about it in the next lecture, and its ability to affect the glucose side of things, okay? Photobiomodulation has been studied all over the place. It started, I think, with pain, and it can affect all kinds of things like inflammation and a variety of things. They're using it for Alzheimer's, they're using it for Parkinson's, they're using it for all kinds of different conditions, okay? Heart disease, heart failure, you name it, because it's all a mitochondrial problem. But the point I'm trying to make is that it's also been used in the field of diabetes and insulin resistance and hyperglycemia, okay? And it's been shown to have a large effect acutely and over periods of time of using red light therapy to help control glucose. And I want to dig into that research in a future video. That being said, there's a great question. First of all, there's a lot of great comments, okay, and I appreciate the comments. So someone asked, Dr. P, how can I contact you via email? You can contact me via email because I put my email in the description of every video I publish, so you can reach out to us there. Someone also asked, can red light therapy be beneficial alone without metabolic therapy? I mean, that's kind of what we've been talking about this entire video, right? Like, this is looking at photobiomodulation basically in a vacuum. They're not using photobiomodulation in keto. They're not using photobiomodulation in fasting. They're not using photobiomodulation and HBOT or IV vitamin C or anything like that, or whatever other thing we use for metabolic therapy. They're using red light therapy alone and showing benefit. So yes, it can have beneficial effects alone. But I ask you, why would you want to do it alone? Like, if you have the ability to synergize it in with what we already know has biologic sense and efficacy with Dr. Seyfried's work, and it makes sense that it would dovetail nicely into that, why would we not want to use that? Let me see if I can look for a couple other questions while. It sounds like you're doing a lot. This Axis is saying that they're fasting, red light, methylene blue, lipoic acid, ivermectin, Finben, deuterium depletion, water, and grounding. So one thing I would say is that, and this is why I want to have a separate video on methylene blue and photodynamic therapy, because it does matter the route of administration and the dose of those kind of things. And so therefore, I really want to do a deep dive on that. I'm not saying you're getting no benefit at all with doing methylene blue orally, but you're probably not getting a whole lot of benefit, comparatively speaking. Yeah, I know. You're asking great questions. Turmeric is a photosensitizer, and I wonder if that helps with metabolic issues. Well, we know turmeric has effects on multiple pathways, probably more than 50 pathways, including glucose and glutamine. So it definitely has effects on the mitochondrial things and the metabolic part of things. But the question is, does it have a synergistic effect with red light therapy? Or I believe, if not mistaken, from listening to an old Dr. Cruz lecture, I think that turmeric actually absorbs in the ultraviolet range. So the question is, would ultraviolet photobiomodulation, or as I always recommend, getting outside in the sun as being the primary way of getting light therapy, would that synergize with turmeric? And the answer is, I don't know. I think it should be studied. That's for darn sure. Looking for other. UW, where Dr. Pollock is doing photobiomodulation for cancer, is showing as much efficacy as radiation and proton therapies. I would not doubt it at all. I mean, mechanistically, how does proton and those kind of things work? It acts like radiation therapy. It acts with as overwhelming amounts of reactive oxygen species. That's why it makes more sense to use the same mechanisms as things that we know work, like radiation, but use modalities of delivering them in a way that is synergistic with the body, not detrimental to the body. So red light therapy, IV vitamin C, HBOT, ozone, those kind of things like that we know can likely not harm or even help the surrounding tissues, but don't have the side effects of radiation-induced fibrosis and radiation. I mean, there's just so many different problems with radiation. This is a funny comment. Brother, I'm actually following you. And the bottom line is, if I'm following you, damn near everyone else has got to be following you. LOL. Yeah. So I appreciate everyone following. Yeah. Look at this. Someone's putting sulforaphane absorbs at 570 nanometers. The only issue is with that, Christopher, is that, a lot of the devices don't have those frequencies, right? It'd have to be like a visible bulb, like a more full spectrum visible bulb, like an incandescent, or a special LED that is at 570, because most of the PBM is going to be in the 600 nanometer range and the 800 nanometer range, which is where the majority of the studies are done. Although there's studies all the way up to 1200 nanometers showing benefits. So Yeah, that's the only issue. Old street lamps. Yeah, I mean, I think so. Part of the thing that frustrates me, and I appreciate people asking the questions, is that like they want to know like you know absorption of things. They want to know dosing, scheduling. They want to know certain things that I can't answer. Like I can only use the literature we have, right? I can speculate. I can give you my opinion, but I can't say for certain. I mean, the work has to be done with people who are researchers, people who are at the bench, people who are doing the work. I'm not a researcher, you guys. Like I'm not. I mean, I work in a hospital. I do functional medicine consultations. I don't do I do I'm a patient care person. I don't even want to go to research. I have no interest in that. I didn't like organic chemistry lab when I was in college. Why would I like it now? So but we do need people who are out there, doing it, like the Gerald Pollocks, like the Alexis Cowens, who Jack is helping out. I think getting some funding so they can do some of the studies that they want to do on like deuterium depleted water with photobiomodulation and looking at the EZ and how it changes, because there's, I'll digress. Dr. Cruz, he would know much more than me. They believe there's a much different effect, or a much more exacerbated effect, when you use red light therapy or you use photobiomodulation on water that is deuterium depleted, because everybody's using tap water. Would a 155 parts per million water be much different than 10 parts per million or 5 parts per million? Dr. Cruz's belief is that yes, because it changes the density of water and it's going to affect the things that I don't quite understand. But my point is that there are researchers out there who are doing it. Yeah, dosage is key. I agree. And, I think it's real interesting that we're also using the dose response curve against cancer cells. So for those who know about PBM or people who don't know about PBM, like I want to reiterate, like this has been studied for like 20 plus years, right? Probably since like the 60s actually, but like really heavily over the last 20 years. Michael Hamlin is like the guy within the space, although I think he retired. He was at the like Harvard or I think Harvard's like light medicine like center or something like that. And he has books and stuff like that as well. But yeah, and the point is that what you may see as a downside of red light therapy, because you're limited by a dose response for the healthy cells, may be something you can actually leverage for the cancer cells, and you can actually like cause an exaggerated reactive oxygen species response within cancer cells, which I think is pretty cool. Any light outside all day, even better than you hit on the head, Braybouf, Garcia, like hit it on the head completely. Any light outside all day, even overcast and cloudy, is better than nothing at all, 100%. And, even though PBM is amazing and red light, therapy and infrared saunas and Spurdy vitamin D lamps, they're all amazing supplements. They should never take place as being like your only source of light. Like being outside is so critical. Like I cannot underscore enough. Like, I don't even think I'll be able to like get into a lot of the mechanisms of how light, especially like the UV spectrums, like UVA, affects so many different chemicals in our body that are for our benefit, whether it be dopamine, whether it be serotonin, whether it be POMC, the entire POMC cleavage product, gene products. I mean, it's almost unbelievable how it affects, hunger signals, you just name it. Like light is so much more powerful than we understand. And we're really just scratching the surface. We're just starting to like really start to get some, good information about red light therapy. But, who knows, like what other wavelengths within the red and infrared spectrum also have efficacy to a greater degree, who knows, or even other wavelengths, like in some of the studies, they were using blue light. Well, blue light, we generally kind of demonize as being bad, but why is it potentially bad? Is because if we're under only blue light spectrums and we're inhibiting our melatonin, and we're actually, one of the things that I have not shown because we haven't got to that part of the series is that blue light will actually inhibit, or not inhibit, it will increase reactive oxygen species to a detrimental effect. So why not use blue light maybe directly over the cancer cells, or maybe blue and red light over the cancer cells, and then over the rest of the body use red light as a protective mechanism or something like that. Like there's just ways that light has not been able to be studied or leveraged to the degree that I think we will have in another 10 years. Now, I know that doesn't help people who have cancer now and have, a problem now. That I get. You can use the information we have and still get a benefit. It's just that I do believe we're really scratching the surfaces with red light, or light in general, and how it affects biology. All right. Well, we've gone an hour, and I think we've covered a lot today. I do look forward to, in the next video, talking about red light's effect on blood glucose, A1C, fasting insulins, all this kind of stuff in the next paper, because it has direct effects on your GKI, right? Like that's how before doing this deep dive on red light therapy, or another deep dive on red light therapy, I've done dozens of deep dives on red light therapy in the past, but in particular for cancer, and particularly wanted to answer the questions, or at least start to answer the questions about how the studies that show red light therapy not helping anything probably are the exception, not the rule, where the rule is probably it's going to help you. But I'll digress. I want to talk about how red light therapy can be implemented to anyone's metabolic therapy treatment as a way to lower blood glucose and as a way to improve your GKI without doing anything different. Okay? Because to be honest with you, blood glucose is the bane of most of our existence within the metabolic approach to cancer. Is getting it below that average of 80 milligrams per deciliter. Getting your A1C down, getting your insulin down, getting that GKI down is very difficult. And people can only fast for so long without having detrimental effects, especially if you're a very thin person already, and maybe in a weakened state by either the cancer itself or the treatments that we use to kill cancer. And fasting may not be a good idea, or a good strategy for that person. Maybe a ketogenic diet for a certain person is very difficult to follow. Maybe this is a way that we can have you have a little more flexibility with your diet, or if you are very strict, having even more of an effect for you of lowering that blood glucose even further to allow the other therapies that are critical for mitochondrial metabolic therapy to be effective, to have even more of an effect. And so you're getting maximum efficacy from your metabolic therapies. So I do believe photobiomodulation does dovetail nicely into metabolic therapy and should be used as a combination, not standalone, although these studies do show that it has an effect by itself, which is pretty cool. Someone asked, I ordered my Spurdy and looking hard at red light panel. I don't want to commercialize the channel, but I do really only recommend two brands. I recommend EMR Tech, and I recommend Chroma. And the only reason I have a problem with Chroma is just because their devices are so small and they're kind of expensive for how small they are, if you want me to be honest with you. I do think Chroma has a lot of other cool options. Like I have the Sky Portal above me. Like I think it's a great light solution for indoor lighting. But I do prefer the panels. I would say EMR Tech. And for clients, I don't recommend anything less than the Firestorm or the Inferno. Now, I don't think you have to buy like the giant panels. I think that's, if you have the disposable income, that could be beneficial. But the Inferno or the other one, the smaller one, is the only ones I recommend, just because they have the power. It's called the gosh darn it, I can never remember that one. I can always remember the Inferno, but I can never remember the other one. EMR Tech. I'm probably going to think about it while I'm pulling up the website. Firestorm. I don't know. I can never remember the Firestorm name. But anyway, the Firestorm. Yeah, the Firestorm. Yes, that's the one I couldn't remember. Firestorm or the Inferno, just because they have I think they're both north of 1,000 watts. I don't want to say I made the mistake, but I actually bought a giant I actually have 2 panels, pretty big panels from other companies. And I really wish I would have waited, if you want me to be honest with you, until I knew more about red light therapy and had studied a little bit more under Dr. Cruz before I got those. I'm not saying they're worthless, but I would just if I was to redo it, if I was looking into photobiomodulation, I would go with those 2 brands. And I personally probably would pick the EMR Tech over the Chroma, just because they don't have big lights. Yeah, someone asked, Spurdy ultraviolet light machine from Amazon good? So that's cool. They sell on Amazon. They've been well known off of Amazon for, I think, since the 70s or something like that, since the first ultraviolet light they ever made. But yeah, I think Spurdy's excellent. I think a Spurdy and like an Inferno or a Firestorm from EMR Tech would be probably the best combination. I don't like using ultraviolet personally without red light therapy, just because it's never seemed like that in nature. And there's more potential for, overdoing it and harm. So I like red light with ultraviolet. And I think the combination of the Spurdy plus the red light panels from EMR Tech would be good. EMR Tech now makes the Krypton series, but if anybody's looked at those, they're ridiculous expensive. And so I don't generally recommend those, just because it's so out of most people's budgets that I don't think it's not a good idea. If they made them, maybe slightly more expensive than the red light panels, I would say that seems like an easy one-stop shop, but they're not. They're like double or triple the price. So I don't think I can, in good faith, recommend that. Okay, well, let's talk about red light therapy and how it affects blood glucose and glycemic control, and how it can improve your GKI and your metabolic therapy as a whole next time.